So it's my great pleasure now to introduce Dr. John Tsang from the National Institutes of Allergy and Infectious Disease, and he's going to talk about systems immunology of COVID-19, integrating patient and single cell variations.
Yeah, thank you so much for having me, and I would like to thank the organizer for inviting me to come here. So I am a systems immunologist. Recently, together with my colleagues, been delving into COVID during the past more than a year now. So, so today I'm going to talk to you about both our work in adult COVID, but also some of the more recent work on MISC and also pediatric COVID with my colleagues. So first, I'd just like to generically thank my lab and also the Center for Human Immunology, who contributed to the work I'm going to tell you, and I'm going to acknowledge folks along the way. So just a quick introduction on what we do. So my lab is really obsessed with variations at several levels. At the highest level, it's really differences across human individuals. And then at the sort of the lower level, understanding differences across single cells. So we've been really excited in the past several years with technologies coming along that we can really start to explore how different factors can drive human variations in response to vaccines, infections, and diseases. And we can now look at this at the really single cell level, for example, looking at cell population specific differences, examining time trajectories, and also really trying to understand whether cell-to-cell variations and sometimes like folks would call that biological noise would play a role in some of these differences in response to disease. And so, of course, in humans, really it's leveraging the variation across individuals, allow us to learn new biology and ask novel questions. And so broadly speaking, we work on several areas in human immunology. One is I mentioned already, and trying to understand why individuals mount variable immune response to infections, vaccination, and disease. We also try to look at a high level across the board in both multiple diseases, for example, monogenic lesions, and comparing that to the healthy immune system. And to do that, we've been also building tools to look into these directions. The other sort of larger areas we work on in single cell biology and try to understand this question of whether these single cell variations actually make a difference. But I'm not going to go into that today in detail, but you're welcome to discuss that later. So one of the workhorses of our work, it's been single cell profiling. And one of the approaches we've been using a lot in COVID work, it's CyTOF. So CyTOF, it's allow us to profile both the surface proteins plus the transcriptome, and if we want, also VDJ sequences like TCR, BCR sequences at the same time in single cells. So very much like flow cytometry, instead of labeling antibodies with fluorochromes, you can label them with oligobarcodes. And then you can use sequencing as a readout of the level of the proteins. So what we have done, it's really trying to develop a multiplexing approach to allow us to look at individuals over space and time. So here I'm highlighting, we're looking at patients, and we can collect samples over time. And we try to barcode individual donors by using their genetic variations. And also we try to barcode the time points by using antibody hashing barcodes or also lipid-based barcodes that allow us to really multiplex together over 40 samples and process all of them together. So for example, if we want to sort them, specific cell populations, we can do so after pooling. So with that, the kind of data we've been generating involved using panels targeting about 188 surface proteins. As I mentioned, we can get a transcriptome plus the VDJ sequencing at the same time. And the data I'm going to show you, we have done both unsorted, sort of total PBMC profiling, as well as after we pool, we also sorted specific populations like memory T and B cells to enrich for clonally expanded cells. And then that's followed by sequencing to look at these at the single cell level. So on average, for every single sample, we target about 8,000 single cells or so from these samples. So the protein expression data, once you sort of get it out from the sequencing from the raw data, looks a bit like this. You can sort of look at the classical 2D gates. So they do look like flow data, but we also have developed approaches to sort of examine the noise sources in this kind of unique protein expression data coming from the CyTOF. And as you can see, after you sort of denoise and normalize across the single cells, these now look a lot like, really, you can start to even do them, treat them as flow data and start to gate them. But of course, with so many markers, you don't really need to gate them sort of like doing manual gating one at a time. You can sort of do unbiased analyses. So here's one example where we sort of cluster all the cells using all the proteins. So each column here, it's a protein, and the rows are the different cell populations. And you can also cluster your cells at different resolutions. So for example, at the highest level, you can sort of see these major cell types, but then you can sort of cluster them further to look at finer subsets as well as states like activation. And then once you cluster these cells, you can start to look into the transcriptome, for example, within specific cell populations. So this is sort of one sort of high-level conceptual slide on some of how we sort of think about this kind of data, right? So typically we get a population of individuals, and within each person, we have these populations. As I mentioned, you can cluster them at a single cell level, and you can zoom all the way into the single cell level and look at their variations. And of course, there's the individual. In this case, for example, if you have a COVID patient, we may have samples that's going over time. So you have variation due to time. That's one other component. And then there may be a perturbation. For example, if you vaccinate these individuals, or there may be an onset of disease, that's another well-defined sort of time point. And then you can look at that after the perturbation. So this kind of data naturally exists in multiple levels, right? So you have time, you have individuals, and you have like basically stable individual differences over time that may be driven more by genetics, for example. So we can start to build quantitative models on the variation, these parameters. For example, we can ask, how does the expression level of a protein or a gene within a specific cell population vary? And does that depend on things like age or the specific perturbation like a disease that we're studying? So we can start to take this kind of data and model it in this kind of hierarchical manner and decompose whether something like age or the disease or the time may contribute to the differences that we see. So I'm not going to go into the technical details, but start to go into COVID and highlight some of the results we have been seeing. And before I go into that, I just want to again acknowledge what I'm going to tell you really took a village, and it's fantastic collaboration with the NIAID COVID-19 consortium put together by Gigi and Helen. So the work I'm going to tell you was primarily led by folks in my lab, so Ken Liu, Andrew Martins, William Lau, and Nick Rachmaninoff, and with contributions from many others in NIH, and also with samples coming from Italy. So COVID-19 is really an exemplar, as you will appreciate, of variation, both in terms of its manifestation across individuals, as well as how the disease evolved over time. So first, I would highlight our work in adults to sort of illustrate some of the principles, and then I'll go into the pediatric work. So this is recently published a couple months ago, so you're welcome to look up the details. And so I'll go through the highlighted results relatively quickly. You can also go onto this website and browse the single cell data. And thanks to the CTSI-CN host, they really done a nice job of hosting the single cell data. So in this study, we look at 33 patients, most of them fairly severe. For most of them, we have at least, for all of them actually, we have at least 2 time points, and one that's very early, close to the time of admission. And then we also analyze 14 age and sex match healthy controls. And one thing we noticed early on was that when we look at some of the lab and cytokine-based parameters in these patients, at the time point that's closest to their admissions, we saw that while the most severe cases, like if you look at these critical disease patients or the more moderate individuals, they look relatively homogeneous. But when you look at the critical patients who recover eventually, but they came in as critical, they have highly heterogeneous profiles. So then we thought we want to maybe develop an approach to break the tie and develop a more quantitative disease severity metric to look at these individuals. So we then develop a machine learning technique to ask which one of these parameters may allow us to separate these categories. And so we found these parameters to be the most useful. So for example, IL-6, the neutrophil to lymphocyte ratio and lymphocyte count. So these allow us to accurately delineate these 4 categories. But at the same time, they also provided a continuous measure of disease severity in this group of patients. They're all coming in as critical or ordinal score 6, but they differ based on these molecular profiles. So we then try to validate these in an independent set of patients. So this is looking at an independent set of 67 patients. And again, we confirmed that by computing this metric, this disease severity metric score, DSM, this score differ across these categories in this set of patients. And the most interesting to us was that when we look at their ICU admission status, the DSM score is predictive of that. So when you look at the whole set of patients, the patients who got admitted to the ICU had a higher DSM score than those who didn't. And more importantly, if you look at the patients who came in as critical, we can also differentiate between those who got admitted to the ICU versus not. So this information, again, was not used in the training. So we only trained this whole model using the initial 33 patients that we got for our CTSI-CN analysis. So this is reassuring. And now we have applied this to additional cohorts and found this DSM metric to be very useful. So now we look at the single cell data in that cohort where we have CTSI-CN. So this is a very high level summary of the status of the cell populations. So on the left here, each column, it's a cell population cluster based on the single cells. And each row corresponds to the status or the state of those cell populations. For example, if you look at genes enriched for type 1 interferon and antiviral responses, you do see that compared to healthy controls, the COVID patients are significantly elevated in that regard across most of the cell types. But when you look at its association with disease severity, you do see that's also now been reported by multiple other studies that the more severe the patient, the lower the type 1 interferon score. This is even after you accounted for timing and age and gender. So timing is a very critical variable. So we do see that over time, the type 1 interferon signature drops over time, even for the milder patients. And the other aspect is that you do see that inflammatory signatures, it's quite elevated across the board when you compare the healthy controls. But again, paradoxically, when you look at the more severe patients, inflammation actually, it's attenuated. So the other aspect we look at is to look at what are the correlates of disease severity, right? So when you look at that, you actually can find tons of variables that are just correlated with disease severity when you look at them across patients. So what we did is we developed an approach to try to, this is a computational approach, and try to fish out the most critical variables. So these are variables that are correlated with the disease severity, and their correlation cannot be explained away by other variables. So in essence, we call them the primary or the direct disease correlates. So what this analysis revealed were sort of these 4 direct correlates. One involves apoptosis or sort of oxidative stress signature in PDCs. So I'm going to actually show you the details here in this slide. And the other one all involve 56-dim CD16 high NK cells. And these all together reflect 2 processes. One, it's an elevation in fatty acid metabolism genes. So these are positively associated with disease severity. While inflammation in these cells, as reflected by these gene sets, they are down, as I showed you before, broadly speaking. So this apoptosis signature is quite interesting in that in blood, when you look at PDCs, their levels are lower in COVID, and they also tend to go down as a function of disease severity. And so this might help explain why these cells are lower, because they may be dying. There's also other data from others suggesting that these cells are not really migrating into tissues, because they also found that PDCs are also low in tissues. So if you drill down further into the NK cell signature, one thing that's very intriguing is that IL-15 in circulation is strongly associated with the disease severity. So the most severe patients, they tend to have elevated IL-15. And IL-15, as we know, it's a very early acute activator of NK cells. So in a way, these cells may be behaving in a strange way because they've been exposed for quite a long time to very elevated levels of IL-15. As you see here, IL-15 is also associated with the fatty acid metabolism signature I mentioned. And paradoxically, despite the elevation of IL-15, the inflammatory signatures, including the level of interferon gamma, it's lower in the most severe patients. And finally, in these adults, one thing we noticed was that looking at the later time points around the days 17 to 23 after symptom onset, there's this kind of flip. So what happened was that in the more severe patients, we see that while their inflammation signatures seems to be lower before day 17, but we look at after day 17, they start to go higher compared to the milder patients. And you see that both in monocytes as well in these NK cells. And we sort of confirm that also by looking at circulating cytokines. If you look at IL-6, the reason why this is the case is that IL-6 sort of stays high in this case, while they are becoming lower in the milder patients. While in some other cytokines like TNF related secreted products and IL-13, there may even be an elevation in inflammation, while the milder patients are starting to come down. So to validate this concept further, we sort of put together another sort of set of a cohort independent of the CTSI-CN cohort that we had done, where Gigi and others had already obtained circulating protein measurements. So in this one, we specifically assembled sort of 21 recover patients, and also 17 that were deceased, with the idea of trying to understand whether there's a divergence around this time point between these two sets of outcomes. And indeed, when we look at that around this period, we found a significant number of markers that differentiate between these two groups, IL-6, IP10, IL-17, and also LBP, as well as certain maybe barrier and tissue inflammatory products are found that some of them even go up. For example, lipocalin, by day 17 or so, they start to ramp up in these diseased patients, while they stay sort of flat in the recovery individuals. So this actually allows us to predict fairly accurately who's going to survive and who's going to die by this period. And this also provides some insights into what may be going on in some of these patients and why they may become fatal. So these also raise some interesting questions. So really, what is driving that early inflammatory attenuation in the most severe patients would actually, like, for example, targeting IL-15 would make a good target. And also what's driving this potential second wave. And this has been seen actually, but with timing wise earlier in severe influenza infections by a group in the UK. So there may be, for example, some may be due to secondary infections, some may be due to barrier damage, and there may be leakiness. And there may also be viral load related reasons why this is the case. And the other question is whether this kind of early attenuation, especially in some of the innate cells, I call that kind of the burnout state, might be insufficient to handle this kind of second wave in these patients. So with that, I just want to now quickly turn to pediatric COVID and MIS-C. This is ongoing work led by Ken Liu in the lab together with Octavia in Gigi's lab. So in this case, we have applied the same approach to look at 7 MIS-C patients and 8 pediatric COVIDs together with pediatric healthy controls. So this is the distribution on the lower left, the sort of day since symptom onset. So for MIS-C, it's the day since they have MIS-C symptoms, not from the time of COVID. Most of them have asymptomatic COVID infections. So we apply the same single cell approach to look at these. So when we look at their sort of transcriptional signature across the different cell subsets, you see that there's again a fairly broad elevation in interferon signatures, in particular in B cells and NAIT cells. Compared to adult COVID, we see the opposite in that these samples had elevations in antigen presentation genes. Often in adults, we see the opposite. We see a repression of antigen presentation genes, which is coupled to the attenuation I mentioned, the inflammatory attenuation. And you see these very strong T and B cell activation signatures in the lymphocytes. When we look at pediatric COVID versus healthy, we see even stronger interferon signature and also antigen presentation. And as I mentioned, I'm showing you in the bottom sort of a contrast with the adults. So as you go to the least severe patients denoted as PC1 low, all the way to the most severe patients, you see the antigen presentation genes, they tend to go down. So when you now compare MIC and pediatric COVID, you see that quantitatively speaking, the interferon signatures are lower in these MISC subjects compared to the pediatric controls, and same thing with antigen presentation. So likely because the pediatric COVID patients actually are mounting a fairly robust response, a robust innate response compared to the adults, and also compared to maybe the different kinds of inflammation in the MIS-C patients. And I also want to point out that this may also be because of the fact that these patients had steroids. We do see that steroid response signatures are up in some of the cells. And so overall, we see sort of this kind of lower inflammatory signature, higher type 1 interferon. But interestingly, when you look at the inflammation, for example, genes due to NF-kappa B, you can see that in the pediatric COVID, they're actually lower compared to the adults, basically. So in adults, in some of them have even more elevated inflammation. So finally, I just want to conclude by pointing you to some interesting observation on B cells. So Can noticed that the frequency of memory B cells seems to be quite high in these MIS-C samples. So basically what she did is she then further clustered just the B cells using all the surface markers. And one cluster that was quite distinct that she noticed was this cluster 8, which is CD11C high. And these cells are CD11C high and TRAF5 low in the RNA, and also 21 low and 27 low. And these are been described before in lupus, for example. These are the sort of extrafollicular B cells. And so it's quite interesting that they are showing up here, and when you look at them and they confirm what's been found before, that the genes that are expressed in these cells are highly enriched for genes like that's been linked to lupus and diabetes and other autoimmune diseases. And their frequencies also tend to be enriched in the MIS-C patients compared to the COVID and healthy controls. The other aspect that Can noticed was that the mutation frequencies, when we look at the VDJs, are also higher in some of the B cell subsets, but not exactly in this CD11C high subset, but in the other memory subsets like cluster 1, 2, and 9. So she found that when you look at them in memory B cells or in plasmablasts, the mutational frequency is significantly higher. And those tend to be markers that are denoting activated cells. So she correlated the surface markers of these cells to the mutational frequency at the single cell level, and found that things like CD95 and 27 and CD71, for example, are associated, suggest that these more activated cells also carries with them significantly higher mutations. So just to summarize, we thought that perhaps the virus-induced the elevated interferons in BCs may trigger a loop that's often been sort of hypothesized to be responsible for autoimmunity in diseases like lupus. I'm happy to discuss that in the discussion. With that, I just want to thank again my lab and other collaborators, and thank you for your attention.